Okay, everything looks good. Great. Okay. Yeah, thank you for that introduction, Nikolai. And this has been a really great conference. Thank you for putting everything together. It's really too bad we couldn't see each other in person, but I think the discussion has been really involved, which is really nice for one of these virtual conferences. So today I'm going to be talking about some of the work that actually started after attending the very first single cell conference in Boston about 3 years ago. And this started some experiments in my lab really trying to understand the effect of the carrier proteome within these multiplexed experiments and how that might affect the data that we're getting from these types of approaches. So I'm kind of highlighting here, obviously everyone within this conference is very familiar with single cell proteomics, why we'd like to do it. This was a very nice piece about single cell proteomics, and it highlighted a few different technologies. There were some technologies basically that have reached the single cell level. There were things like mass cytometry, fluorescence imaging. There's some techniques that have a lot of promise, and I think Ed Marcotte was actually in the audience, so this came out of his lab, combining single molecule fluorescence as well as Edman degradation. But I think the biggest part about this piece was that there was a lot of discussion about the work that Nikolai and Bogdan did around multiplex proteomics using isobaric labels. And what this demonstrated was that these approaches have really kind of entered the mainstream and been introduced to a broader community as an approach towards analyzing proteomes of single cells. So obviously, most of you are going to be familiar with the TMT labeling approach or isobaric label-based approach. I won't go through the actual workflow, but I wanted to summarize some of the advantages and challenges. Of course, we know that with multiplexing, you can get multiple samples in one analysis. Typically, you're going to have limited missing values. However, with single-cell proteomics, that's not always the case. The additive peptide signal is really what the carrier proteome brings to the table, and I'll talk more about that, but it's basically just that once peptides are labeled and mixed, their signals add up in these survey scans, and that's an important factor to the carrier proteome. Some of the challenges include things that have been well studied, including precursor interference, the fact that TMT labeling requires more advanced sample manipulation, and I think there's been a lot of great talks at this conference in terms of improving sample manipulation and sample handling for these single cell samples. Now, coalescence is a concern that has been raised with TMT, and traditionally I hadn't talked about it, but I'm actually going to talk about coalescence some today as well, based on some of the data we saw with really high carrier proteomes. And lastly, there's ion sampling, and this was really, ion sampling was kind of the first concern I raised about this carrier proteome approach when we first met 3 years ago. And since then, other people have looked into it too. Actually, there was a paper from the PNNL group published in MCP earlier this year that had a lot of the same messages I'll talk to you about today. So if you're interested in what I have to say today, you can head over there pretty quickly and see some data that will, data that basically agrees, our data agrees with each other quite well. So, of course, SCOPE-MS is why we're here. This is the paper from Bogdan Nikolai. And I'm really highlighting this because if you look across the field, basically with experiments that have been published, experiments that have been talked about in conferences, things on preprint servers, the carrier proteome typically is spiked into samples at around 25 to 500 times the single cell proteomes. Now, when you have that type of situation, what you're going to see is survey scans that look like this, where the majority of the population is coming from the carrier proteome, not only in the survey scan, but also in the fragment ions as well. And that's something that we have to keep in mind as we're using the fragment ions for detection and identification, is that these are mainly coming from the carrier proteome ions. We then do either within the MS2 or if people want to do in the MS3, quantitation based on a reporter region that has kind of a very small amount of signal coming from those single cell lines, and then a more prominent signal coming from the carrier channels, both the actual carrier channel itself as well as the natural C13 isotope. So I'm going to talk about two challenges here today. One is going to be the challenge of actually collecting enough total ions so that you have enough of these smaller ions to quantify these single cell ions, as well as the challenge of actually just measuring and manipulating ion clouds or ions within the mass spectrometer when you have a very large population combined with another smaller population. So I'm first going to start with this sampling idea. And Nikolai did a really nice job of introducing this idea about sampling, increasing AGC targets at max inject times. I'll be talking more about that. This was a visualization I put together that I think helps kind of underscore the idea. So these are 4 different ion clouds. Each of these ion clouds actually contain 1,000 ions. And within these ion clouds, we have different carrier levels. So the carrier proteome could be spiked in at a 5 times, which would mean there would be 5 times as many gray dots as blue dots, and then just as many color of each of the colors as one another. So those are all spiked in at 1 to 1. So you can increase the carrier proteome, and as you do that, obviously, if the number of ions is constrained, what you see is that you actually get fewer and fewer single cell ions actually represented in the cloud. So if you take a small sampling of these ions, what you see is kind of the first effect, that at a particular sampling level, if you increase the carrier proteome, kind of for a defined number of ions, you're actually going to be increasing the variation. And this is the variation over 100 different simulations of this random distribution of ions. Now, you can overcome this, obviously, by sampling more ions. And as you sample more and more of the ion cloud, of course, you get more and more of the representative population, and your variation on your measurements decreases quite a bit. Now, even though it decreases to where they might be all somewhat equivalent, you see there's always, basically at each sampling level, going to be some dependence on the carrier proteome. If you have more carrier proteome, then you're not going to be sampling as many single cell ions. So you're going to have an effect where you're going to get more variation with higher carrier proteome. So I want to talk then about some experiments where we're going to be talking about sampling enough ions or detecting ions. So to do that, I'm going to be using a measurement that we call signal to noise. So this is something you can get off of the instrument, really any thermal Orbitrap instrument. If you look at the reporter ions within a spectrum, you'll get a couple of values. You have the intensity. That's kind of the intensity that a lot of people use for the analysis. But you also get this noise level. And if you notice, the noise level actually increases slightly as the M over Z increases. So if you do this ratio of the signal to noise, you actually get a value that's roughly proportional to the number of ions. Now, exactly how that conversion happens is actually somewhat variable. It's anywhere from about 2 to 5 ions per signal to noise unit. And that's going to be dependent on the type of instrument you have, the length of transient you're using, the resolution you're detecting at, and everything like that. If you want to learn more about this, there's a really great MCP paper by Martin Wer's group from Princeton cited here, and I would definitely point you to there. So I'm going to be using signal to noise as a proxy for the number of ions. That's just shown here. I'm also going to be talking about this idea of ion flux, and it's really this idea of how many ions can I get per millisecond injection time. And this is really important because we want to know how long we need to inject to accumulate enough ions to get a good spectrum. So just to kind of orient ourselves on what we might be looking at in terms of the signal to noise range we might expect from single cell experiments, I analyzed some publicly available data. This included this paper that utilized nanopots from 2019, as well as the SCOPE 2 data. This was actually the first version of the SCOPE 2 data that used TMT10-plex. And I looked at the signal to noise within all identified spectra. So I actually took the data, searched it, identified PSMs, looked at the signal to noise distributions within those spectra to see where we landed. And you can see this ranges from about 500 signal to noise as the median when you use a carrier of about 25x to about 2,000 signal to noise units as a median when you're using 250 times carrier. So it will be somewhat dependent on carrier, but we can kind of think about the signal to noise we're getting within single cell experiments to be somewhere between 500 and 2,000 signal to noise. And that'll be important as we look through the next set of slides. So to kind of look deeper within those kind of trends that we were seeing within the ion cloud simulations, we took a peptide. This was actually a peptide within the Pierce retention time calibration mixture. And we labeled it with TMT10-plex. We have one of the channels spiked in at 50 times. This represents the carrier proteome. Everything else was labeled at 1 to 1. Now we analyzed this with a targeted method, and we did that so that we could actually analyze the peptide with 3 different sampling levels. So we have kind of a low number of ions, a median number of ions, and a very high number of ions. Now 1E5 might be what's more common to people in the field. And as you can see, we got 10 points across the curve so that we could also see the effect of when in the elution period if the peak was sampled and how that might relate to quantitation. Now, if we look at one of these a couple of these spectra. These are the actual signal noise values for the reporter ions when we have kind of a low number of ions. In this case, we have 165 signal to noise units, and you can see that how it's distributed here. What you can see is that we're actually getting kind of sporadic detection of these low level single cell channels, right? These would be those channels that represent single cells within this peptide mixture. It's such that, you know, if you sample at the peak apex, you're actually only detecting 5 of the 8 channels we had in there. If you sample it one scan later with basically the same number of ions, you actually see 7 of the 8 channels. So we're getting really sporadic and kind of random detection of these ions. And this is really the main point I'd like to make with a lot of the ion sampling I'll be talking about today, is that if we do severely undersample ion populations, we're going to be getting sporadic events that may look like cell heterogeneity, digital measurements, but it could just be the fact that we don't have enough ions from the sample to actually measure them. They could actually be present. And we know in the sample that they are, obviously, because we mix them in. So if we sample enough ions, we actually see now that we get reproducible detection of these. You can see that the CVs across them are actually quite good. And in this case, with the 50X carrier, this all happens with about 1,000 signal to noise. Now we looked at how the CV across all of the quantitative channels here relates to where it was sampled in the peak. And it's not really, there's no real clear correlation. And this is really just kind of the AGC target working so that we ask the instrument for a certain number of ions, and here it's kind of delivering a certain number of ions. So the CV kind of plateaus. But we see a really nice relationship when we look at the number of ions measured here as sum signal to noise, including the carrier. So it's all of the sum signal noise of every peak versus the CV. So you can see with low ion sampling, we're getting very variable measurements. If we increase that, we can actually get very reproducible measurements and limit the variability quite a bit. So we expanded this beyond that just one peptide and one carrier amount, and we looked at basically all 13 peptides, and we looked at carrier amounts of 550, 250, and 500. And when we do this, what you can see is that we get what we're defining as the carrier proteome effect. And that's as you add more carrier proteome, you're going to have to increase the number of ions you sample to maintain quantitative accuracy. So if you look at these with a 5x carrier proteome, you're actually getting pretty accurate data kind of no matter where you sample. There might be a cutoff here, but it's going to be very low in terms of how much signal you need to quantify a spectrum. But as you start going up into 50, 250, and 500, you're going to be needing to sample many, many more ions to actually maintain that quantitative accuracy. And at some point, you're actually going to need more ions than you might actually be able to obtain within single cell experiments. And again, that might be somewhere more in this 500 to 2,000 range, what we might be able to get with single cell analyses. And that's where these measurements are going to be more variable if you have higher carrier proteome amounts. Now, we also looked, obviously everything I'm going to show you is really, everything's mixed 1-to-1. So I did just want to say that we do see similar trends with mixed ratio samples. So in this case, we actually took carrier proteomes of 15, 100, 50, 1,000, and 2,000, and we looked at mixtures of 2-to-1 in a very similar system. And here you can see again with a 15x carrier, we don't really see any effect on the measured ratio and the accuracy of the measurement. But when we get up to, in this case, 150 carrier, we actually need about 1,000 total ions, including the carrier, to maintain accuracy. And I left these other points in here just to highlight the fact that when you get up to very, very high levels of carrier proteome, 1,000, 2,000 times, it becomes very difficult to actually accurately measure ratios, no matter what ion sampling level you're at. So the ratios here are actually pretty variable. So very high carrier levels are going to be kind of detrimental to quantitative analysis in this way. So now all of that data was with single peptides again. And what I wanted to do next was actually look at this within complex mixtures. And this was all done with bulk level analyses. There's actually no single cell preparation done within the data I'm going to show you today. And we needed to do that because we wanted to remove two things. One, the cell heterogeneity that might exist within single cells. And two, the differences introduced due to sample preparation. So we needed actually the same exact sample to be mixed together, and that's what we did. So we took the same exact 8-plex sample, which was 4 HeLa cells and 4 K562 cells. This was actually the same exact sample split into different channels. So it was the same lysate. And we took that exact mixture and we diluted it into the carrier proteome background, which was a mixture of HeLa and K562, so that we knew the exact same ratios should be detected within this 8-plex sample, because it was exactly the same sample that was then diluted. And what you see here is exactly what we saw with our peptide measurements. Basically, as the carrier proteome increases, we're going to need more ions to maintain quantitative accuracy. Now, again, if you're using something like 300 carrier proteome, you're going to need something on the order of 3,000 ions to reach a median CV of 20%. And again, that's going to be something that might be a lot higher than what you might be able to obtain in single cell measurements. So in this case, using a carrier level at or above 300 might really challenge your ability to get quantitative data. Now this the middle graph was really based on the total number of ions, but we also wanted to put this in the context of just the single cell ions or the ions in the quantitative channels, right? So if we look at just those, we actually see a pretty nice, a pretty kind of carrier proteome independent relationship here. And that makes sense. You should only need a certain number of single cell ions to actually make this a quantitative comparison. But what we didn't expect is that we were actually starting to see increases in variability as we got more single cell ions. So we saw a nice kind of relationship that there's a certain number of single cell ions you need to reach this 20% CV. But if you got too many, we actually started seeing these offshoots shown here with this 300 times carrier sample. And this was saying as you sample more and more ions, you're getting less and less accuracy, more and more variability. So we wanted to look into why that was the case. And what we saw was something really interesting. So, within this 8-plex experiment, the HeLa channels were actually closest to the carrier proteome. Those were in channels 127N and then all the way through 129N, of course, skipping this 127C, which is the 13C isotope of the carrier. The K562 channels were all on the far right side. So those were all over here. So those are the farthest away from the carrier. So what we see then is that the K562 has a pretty nice distribution until you actually get to later or higher ion sampling amounts, and that could be other sorts of things happening there I could talk about later if you want. But if you zoom in on this lower region where we might expect more single cell analyses to fall, what you see is that this K562 has a nice relationship, whereas this HeLa one actually has these offshoots where we're getting drastic increases in variability as we get more single cell ions. And what we assume this might be is actually coming from this guy right here. So this 127N channel. Now, as you get more and more ions, this 127C channel will actually grow, right? Because as you accumulate more and more ions and you have high carrier levels, this isotope will actually increase as well. And what we believe is happening is that we're actually getting ion coalescence, which is this phenomenon that occurs when two peaks, which are really close, and they're only 6 millidaltons away from each other. When you get a lot of ions within one of those two peaks, or in both of them, they actually come together within the mass spectrometer. So we left that channel out of our analysis, and then we saw that this mostly took away the effect, but it's still somewhat there. And what we think is happening then is actually that the 128C channel from the HeLa is likely being interfered with a little bit with this 13C2 isotope from the carrier. And we've actually seen this channel increase in intensity if we go to very high levels of carrier before, so we think that's the case. So this is just to say that if you have more than 300 times in your carrier proteome, you are going to have to actually be careful of not only that you have enough ions, but also that you don't sample too many ions because you might run into coalescence. You could, of course, get around this by just leaving out those two channels, taking what was an 8-plex down to a 6-plex, but that could be one avenue to get around this. The next kind of big question that came out about the time that the initial paper that Nikolai and Bogdan published was introduced was really, does the Orbitrap or the mass spectrometer really have enough dynamic range to analyze these types of samples? So we made an experiment on the instrument to test this. And the way that we did this is kind of highlighted here, but it really just revolves around fragmenting a peptide and then doing a second round of isolation on just the reporter ions, including the carrier ion, or within the reporter ions excluding the carrier ion. And because I think all of you are very, intuitive mass spectrometrists, you might notice that that last slide was not very accurate. So I like showing this one when I believe people can handle it. And this is really what's going on in the instrument. So here we have the quadrupole. We're isolating a peptide, obviously fragmenting it. And if you're doing just normal MS2 analysis, you would take this, which includes the carrier proteome in gray, the reporter ions, and the fragment ions, and take that to the Orbitrap for analysis. But here what we did is we actually took it into the ion trap as an MS3, and we then isolated either a window that included the carrier, so this was centered on 129.13 and it was 8 Thomson wide, so we included the carrier. Then we shifted that isolation window over about a mass and a half, and we then isolated the reporter ions without the carrier. And this is how we actually then tested whether or not the presence of the carrier was causing limitations, not necessarily in detection within the Orbitrap, but honestly more with the manipulation of the ions from here in the ion trap or the HDD cell through the C-trap and into the Orbitrap. Now, what we saw was really interesting. So with low carrier ion levels at 5 times, you really don't see a difference. But as you start to increase the carrier levels up to 20, 75, 300, you start to see this really big difference between exclude scans, which don't have the carrier ion, and include ones which do. And what's likely happening here is that we're actually getting some space charge effects. Space charge effects are what happens when there's too many ions being manipulated within the mass spectrometer, and you actually end up getting kind of a limitation into the number of ions that you can manipulate with high fidelity. So you're going to lose some ions along the way. And what appears to be happening is that at high amounts of ions, we're actually getting a plateau, which is about 3,000 ions here for the 20X and 75X, and about 300, a little less than that, ions for this 300X carrier. So this just means that even as we accumulate more and more ions, if we have the carrier proteome channel present, we're actually not seeing an increase in terms of the number of single cell channels. And that's what I'm showing here on the y-axis. So this is the single cell channels. Now, obviously, this is kind of rectified by getting rid of the carrier proteome channel by doing the isolation in the ion trap. Now, while this is really interesting, I do want to bring up the fact, if you look at this as a percent increase, basically taking this orange line and dividing by that blue line for every spectrum, what you see is that you actually, within the range of ions that we're typically going to be seeing in single cell proteomics experiments, this really isn't an issue. I think it will be an issue if you use very high levels. I only went to 300 because honestly, I don't think people should use any more than that. But at the levels of single cell proteomics, which might be 500 to 2,000, this really isn't going to be an issue. So I don't think the dynamic range is actually going to be affecting people's data unless they're actually accumulating more than, say, 4,000 signal to noise or 6,000 signal to noise, which I don't think people are going to typically be doing. The last kind of like method development aspect of this we wanted to look at was the level of multiplexing. So obviously the level of multiplexing has kind of been a path forward for single cell proteomics. If we can increase the level of multiplexing, we're going to increase the throughput as well as hopefully be able to decrease the amount of carrier. So what we wanted to show here was basically what's the effect of increasing the multiplexing levels on the accuracy of the data kind of as a function of the carrier proteome. So we looked at, this is a TMT Pro sample, again, with the carrier in the 126C channel. And we have 5X carrier, 15X carrier, 60X carrier, and 250X carrier. And what you can start to see, not too much at 5 and 15X, which kind of makes sense based on everything I've been showing you, but more with 60X and 250X is that you do kind of get this plexing level dependence of how many ions you might need to kind of maintain the quantitative accuracy, or even just, kind of this absolute quantitative accuracy you might be able to achieve based on the level of plexing that you have in your experiment. And this doesn't really have too many implications, I think, for a lot of experiments out there, but it does have implications on something I'll show you later, and that's basically trying to set a quantitative signal to noise threshold, or the number of ions within single cell channels. If you want to set a threshold based on that, we actually have to factor in how many channels we have, because as you have more channels, your single cell signal will be split. So you will need to accumulate more ions to maintain accuracy. I do want to note as well that we do still see here with this 250X carrier that we get this increase in variability as we get more and more single cell ions actually detected. So this is still something that we should keep an eye on, especially when using very large carrier amounts. So this is the last experiment that we did, and it's basically taking two of those samples, either a 20X carrier or the 100X carrier, and taking that same aplex sample and then dividing it or diluting it into single cell levels. So we started with about 400 picograms per channel, and we diluted that all the way from 200 to 100, 50, and 30 picograms per channel. And as you might expect, the signal within the single cell channels decreases as we decrease this amount on column. Now, I also kind of want to highlight this idea of the quantitative ion flux, the idea that obviously because as ions are being accumulated in the mass spectrometer, if you have a lower carrier amount, which is what this 20X is, you're actually going to be accumulating more single cell ions per millisecond of injection time. So the injection times would need to be slightly lower for the 20X than the 100X because of that. Now, to analyze the sample, we did something that was very similar to what the group from PNNL did. But we analyzed this with different instrument parameters. In this case, we looked at low, which was 5E4, 150 millisecond max inject time, common parameters, which was 1E5, 300 millisecond max inject time, and high, which in this case was 5E5 and 750 millisecond max inject time. And we looked at the number of PSMs, which I'm showing here. This is peptide spectral matches. This is basically how many spectra were mapped to peptide sequences. And what you could see is that, kind of as Nikolai was describing in the first his first talk, basically you have different modes you could operate in. You could operate in a mode where you're just trying to optimize the number of proteins you identify, and you could do that with the low parameters. You can see here we get about 1,200 proteins at 400 picograms, or you could actually slow down the instrument and collect more high quality data. And if you do this, you can get something closer to 600 proteins. The real difference here is the number of these changes that actually have CVs less than 20%. And that's what I'm showing here. So obviously, as you look at this data, you'd be very excited because 1,200 proteins is quite a bit. But if you look at the underlying data, you'd see that really only 23% of these actually have a CV less than 20%. And if you look at the distributions of the CVs, you can see that a large proportion of the data is actually not very accurate. And as we're coming to single cell proteomics within my group and at Genentech, we're really trying to ask questions on really a protein by protein basis or on a pathway basis. And we're trying to use proteomics generally within our group to look at kind of mechanistic biology. So from our perspective, kind of the accuracy of the data kind of on a data point by data point level is very important so that we can determine which of these proteins is changing and be confident that we could spend months, years following up on a hit. So what we decided to do then was actually implement a post-analysis threshold for quantitative signal to noise or signal to noise in those single cell channels. And we did that very briefly by just looking at the correlation between, or the relationship between single cell ions and the median CV we looked at before. And what we did was actually set a level where the percentage CV was about 15%, and that ended up being about 125 quantitative signal to noise. Again, this should be dependent on the number of channels you have. That's about 15 quantitative signal to noise per channel. So we basically then filtered out PSMs that did not have that quantitative signal noise level. And as you might imagine, the underlying data improved dramatically. So the distributions of the CVs really improved quite a bit, such that nearly all of the analysis had the majority of the distributions below 20% CV. Obviously, with the 20X carrier, this dropped it down to a very small number of proteins. So the actual variation is based on very few data points, which is why those distributions are so off. Now, the big implication of this, of course, is that you're going to get fewer PSMs and proteins identified. You can see here that the 1,200 protein sample went down to about 250 after we did this filtering. But with the 100X carrier and these high parameters, we really didn't lose that many proteins. Before the analysis, we had about 80% of the data that was actually pretty accurate. After doing the filtering, we actually had about 96. Almost every data point was very, very good. with the 20x, it's kind of interesting because there are situations where you may not need to use these high parameters because you are accumulating more of the single cell ions per millisecond injection time. So that's just showing that there are kind of an interplay, there is an interplay between the instrument parameters, your carrier proteome level, and all of these different things to achieve the types of results that you would need for your particular analysis. Now, I'm going to spend just a brief minute talking about this, and this was really the question of what is the effect of this quant signal-to-noise filtering on the biological interpretation. And I'd say generally all we're doing is getting rid of some noisier measurements. So if you look at the significantly changing proteins between K562 and HeLa, in this case, you can see that generally the number doesn't change too much. All of the data is here on top, the different parameters, the filter data is on the bottom. The number goes up slightly after we filter, but in general, the number of significantly changing proteins shouldn't change too much. And that's really just a factor of the data that we're getting rid of is mostly noisy data, which may not lead to kind of a significantly changing event. In addition to that, we actually did a correlation to not quite a bulk sample, but a sample that was 30 times higher than our 400 picogram sample. So that was a 12.5 nanogram sample at 5x carrier. And after data filtering, so the non-filters is in this first column, after data filtering, we saw just a really increased correlation to the sample that was at a higher level. And of course, this is all the exact same sample being diluted. So the correlation should be very good. So we do see this increased correlation as we filter to the data, which means that we are most likely getting rid of a lot of noise within our data. And that's just shown here for a couple of proteins, where as you can see after filtering, the noise in the HeLa channels actually decreases. And we are then able to get kind of something that matches this higher channel or sample better. So the very last note here is that, you know, throughout doing this whole kind of project, we actually put most of the tools we're using to analyze kind of external data, internal data into a program called SCCP Companion, Single Cell Proteomics Companion. And this is a tool where you can drop in raw data files, you can give it your experimental design, and it'll calculate the quantitative signal to noise, the quantitative ion flux. It'll also figure out things like median MS2 injection time, MS3 injection time if you have it. It'll look at suggested signal to noise cutoffs, so it'll just do a very simple calculation based on the number of samples you have to suggest a cutoff. It'll then actually look in your data and see how much of your data is above that cutoff, which is kind of a nice metric for you to optimize your instrument parameters. And then another cool feature is that we actually suggest maximum injection time. So we take this quantitative ion flux number, which is basically how much of the single cell ion per millisecond you're getting, and we'll actually convert that to a suggested max injection time. And you can see how this varies throughout this dilution series that we did. Basically, as the amount per channel was decreased, we are going to need more and more injection time. And we honestly didn't go up this high. You know, I think at the lowest levels, our program would say you would need somewhere on the order of 2,000 or 3,000 milliseconds, so 2 or 3 seconds to actually get really enough ions to analyze, and that's not something that's going to typically be done, but it might give you a good idea about the sensitivity of your mass spectrometer, which will be really mass spectrometer, sample dependent, and all those different things. We also produce an output which will give you some information about the spectrum. It'll give you all the raw signal to noise values for your raw file. And it'll also give you the reporter ion mass errors. And this is actually something that's kind of a way to look for coalescence in your data. If you see the reporter ion mass errors changing as the some signal to noise increases or the quantitative signal to noise increases, you have a pretty good idea that you are actually seeing ion coalescence in your data, and you're going to need to actually have some sort of signal to noise cutoff. You can see here, this one's actually coalescing because the some signal to noise is about 20,000. That's a very rare event, but it is causing coalescence such that we're actually missing a peak due to that. So with that, basically the lessons learned from this, you can see here we define the carrier proteome effect. That's that increasing carrier proteome levels really requires a concomitant increase in the number of ions sampled to maintain quantitative accuracy. I would still, you know, maintain that 5 to 25 carrier proteome is likely best. I think 100x carrier proteome can work, and I think that really depends on your instrument settings and how long you actually inject for, how many ions you ask for. I really would suggest avoiding carrier proteome levels above 300 times. I think there are going to be a lot of underlying data issues that you may not notice when you're using very high carrier proteome levels. And then I would also say that carrier channels are additive. So if you are using 2 times 200x carriers, that is equal to a 400x carrier. So remember that this is kind of all within the ion cloud, and you have to take that into account. As many others are championing nowadays, AGC targets and max inject times are very important. And I really think that looking at data in terms of signal to noise is really important. Proteome Discovery does this. If you don't have access to that, or you want to use a tool that's a lot faster, you can use SCP Companion to actually look at signal to noise, quantitative signal to noise, suggested cutoffs, suggested max inject times, and all that stuff. So I'd like to thank the people within my group, Tommy Chung, Adam Kiss-McCoy. This work was done in collaboration with Bernard Kuster and Chin Yoon in his lab, as well as Florian. A quick shout out to MS Bioworks. This is a CRO here in the US, but he helped me out with some of these revision experiments because we have been trying to revise this manuscript during COVID, which has been a challenge. But quick shout out to him, and thank you all for your